I'm
Yeah,
sure.
we might not always use the fight if we're legend or not. We always have a big argument about it.
It is a real question.
Yeah. All right, let's get started on this. If you're sleepy, you know what to do. I understand and so forth, but you need to stay awake. So I say I understand, but not really. Okay. So a typical cell. So this whole chapter is about a cell, the cellular level. So we're going to start off with the smaller things at first. Last week we did an introduction to the whole body. This week is the cell. Then we'll move on to the tissues. And then we will move on to the organs and the organ systems and so forth. Okay? So the cell, a typical cell. Cells can be all different shapes and sizes, okay? But they are the smallest but most structural and functional unit of all living matter. The top picture up there, even if you're far away, you can tell that that's something much smaller than the bottom picture. The top picture is a picture of red blood cells, and red blood cells are irregular shaped cells. To me, they kind of look like donuts. This thing says a frisbee, whatever, but they are irregular shaped so that they can move through our arteries and so forth. Bless you. Then the second cell below that is going to be a neuron, and that's a type of cell within the nervous tissue and so forth. So in some nerve cells are going to be about 4 feet long in length as well. So nerves can be all different sizes and so forth. This picture is going to be pretty important to you as you have to label it in your workbook. And there's a little bit of labeling, I believe, on your test from it as well. Not all of it. I will review which ones on Tuesday for you. Yeah, good question, Colton. So, today we're going to start off here after this picture is going to be what the function of each one of these parts is, what the function of the cytoplasm, what the function of the nucleus is, the mitochondria, and so forth. Okay? Let's go ahead and dive into that, and then the later part of the cellular lecture here is more about the pathophysiology of it, or the physiology of it, and the how. Okay, which is a little bit more complicated. So we'll start off here, talk about all the different structures within a cell. Start off with the cell membrane. This is going to be that outer covering or plasma membrane. It separates the intracellular, intra means inside, okay, the intracellular material from the extracellular material. It has a selective permeable membrane that chooses or allows certain substances to cross. So depending on what type of cell it is, depends on what can cross it and the substance that's trying to get across, okay? Like alcohol only crosses certain cell membranes, okay, and so forth. The cell membrane composition itself it's 2 layers. There's the phospholipid bilayer, so that itself is 2 layers. When you see the word lipid, what should you always be thinking about? What's that another word for? Lipids, do you know? Lipids are fats, okay? So this is the fatty lipid. It may not be that you can actually see what you envision fat, but that is what it consists of whenever you see the word lipids. The cell membrane also has proteins in it that provide structural support for the membrane, act as binding sites for hormones as well. It also includes pores that water and electrically charged substances such as sodium and that's not chloride, it's CH. Oh, chlorine maybe. I don't think so either. Cannot penetrate lipid and must use pores. So the area must have pores because sodium cannot get through this cell membrane. A function of the cell membrane is going to be that the membrane itself determines what enters and what leaves the cell. And this is very, very important in many different parts of our life. We think about dehydration and hydration and so forth. Or just like I said, alcohol a while ago, or any other examples of anything, nutrients we take in, sugar, and so forth, that maybe we don't want it to all cross over, and so forth. Also, there are certain drugs that are classified as lipid soluble, so that means that they will penetrate the membrane of the nervous system, so such as the brain and the spinal cord. Right? So those are going to be some bigger ones and so forth. I think everyone probably remembers from elementary or even middle school and maybe high school what the nucleus is, okay? It's like the brain of the cell. Okay? It's like the brain. It controls the workings of the entire cell. It is surrounded by a double-layered nuclear membrane. It contains the genetic information and controls all protein synthesis. Most adult cells have one nucleus, but only mature red blood cells are the only ones that have no nucleus. They operate without a nucleus, okay? But all other adult cells have one nucleus. Within the nucleus is going to be the nucleoplasma and the nucleosis and the chromatin. The chromatin is going to be like the DNA, okay? So the DNA, also known as chromosomes and so forth. That nucleoplasm, that's going to be like the gel-like substance that kind of keeps everything together in there. And then the nucleosis is going to be involved in the synthesis of ribosomes, which we will get more into. So continuing on here, the cytoplasm. So we talked about the nucleus, the cell membrane, and then the cytoplasm is the next thing. The cytoplasm itself, not within the nucleus, is also a gel-like substance. To me, the cytoplasm is like the liquid of the cell, but it's gel-like, okay? But it kind of keeps things floating around in there, keeps them kind of where they belong, and makes it where they could potentially not necessarily have to rub up against each other, but they can without creating friction or problem, okay? This gel-like substance is found inside the cell, but outside the nucleus. It is composed of water, electrolytes, protein, and nutrients, okay? This cytoplasm also contains organelles and cytosol, which includes inclusion bodies and soluble materials like glycogen, granules, and melanin. What is melanin?
Color.
Yeah, color, pigment, okay? That is exactly true. Organelles is the next type or thing, part of a cell. And organelles are little organs, okay? Each one has a specific role, but they are like little organs. I think most of you probably remember, what is the mitochondria? What's the word for it? The powerhouse of the cell. Okay.
By the way, this video is on.
Okay, it's not working at all. Okay. Well, show me a little bit later, like how you're going about it. I wonder why it's not working. And I might have you try to find it on your phone. So the mitochondria is slipper shaped. It is 2 layers. There's a smooth layer and then a folded layer. But the smooth is going to be that outer layer kind of protecting it. And then that inner layer there is going to be those folds or cristae of it. The main thing with the powerhouse, what does the powerhouse create? What's it there for? Thank you. A powerhouse is creating energy. So that's what the mitochondria does too. It uses, it turns nutrients into ATP. And ATP in a cell equals energy. Okay, those words coincide. So the more active the cell, the greater energy output you're going to have, and so forth. So ATP equals energy, and that is what this is doing. All right, then next up is the ER, the endoplasmic reticulum. It is a network of membranes within the cytoplasm. These membranes form channels for moving substances, and then within the ER, there's a smooth and a rough. The rough has ribosomes on the surface, so it's just that. It's rough, really. Okay, it has ribosomes on the surface. Then there's the smooth that has no ribosomes, and it is the site of lipid and steroid synthesis. And next up then are going to be the ribosomes themselves. Okay? The ribosomes themselves are a site of protein synthesis, and some are attached to that ER. And so they might be moving or exporting protein used somewhere else in the body, where others float freely, the ribosomes. I'm not sure, everybody says this one a little bit different. I say Golgi apparatus, some people say Golgi, however you want to say it is fine with me. Okay? The Golgi apparatus puts the finishing touches on the protein after the synthesis begins in the rough ER. And then it packages that protein in the membrane for export or for leaving the cell. So an example of this is going to be where the glucose molecule may be attached to the protein within the Golgi apparatus, and then it helps with transport. A lysosome, this is still continuing on with different parts of the cell that you're potentially going to have to label, right? But a lysosome breaks down waste for housekeeping. So it's kind of breaking down the waste, taking care of the waste product. It has some digestive organelles or digestive organs. We said organelles are little organs, right? So it has some digestive organelles that conduct phagocytosis, which is basically like digesting or eating, okay, through powerful enzymes. So lysosomes, they do the breakdown for the waste. The cytoskeleton, it is very similar, and I love the terminology with it because it is basically the skeleton of the cell. It provides for intracellular shape and support, and it is composed of microfilaments and microtubules. So it really provides that shape and support of the cell. The centrioles are the rod-like shaped structures. They are involved in cellular reproduction, which is very important, and helps separate the chromosomes during mitosis. Do any of you guys know what mitosis is? Mitosis.
It's where the cells reproduce themselves.
Yeah, reproduce themselves. So basically, yes, Reagan. So this is basically the division, okay? So if you think about like one cell, okay, then that divides into two daughter cells, okay? So, and then each one of those divides into two identical cells. And does that process goes in with reproduction that you're talking about. So exactly right. Still talking about the different parts of the cell here. The cilia. Cilia are short hair-like projections where the flagella are thicker and longer, but they both do the same thing, which is enable the cell to move. Okay? The cilia example is very, very important because they should be very abundant in your respiratory tract to help move any mucus and dirt along, okay, or any infection or mucus or dirt. So what happens with smokers is tobacco and the process there ruins or destructs the cilia that line the respiratory tract, so that they don't get that stuff brought up as much, and therefore they have trouble moving that mucus and dirt out of their lungs. These next several slides here are a graph from your textbook, and they're going over the same thing I just have for the past 5 minutes. The different parts to the cell and then the function of each. So I'm not going to repeat myself. This is the next several slides here. So we went through all these different parts and what their function were. This is the video that I'm not going to show you because it didn't like to work earlier. It was about the cell, basically, and describing all those parts and the functions that I just did. Okay? So this is when the tougher stuff comes along here, and this is going to be the how, the why, and so forth, more the physiology part of the cellular structure, how it works, and so forth. Okay? So, movement across the cell membrane. So the cell membrane, that's the outer covering, right? That's the protective layer. And I told you that it's like semi-permeable. It's made up of that phospholipid layer, phospholipid layer. So it's 2 layers. And so there's a couple different ways. You can have either passive transport or active transport of things getting through. Okay? So passive transport, these are going to be movement across the cell membrane. Passive transport require no input of energy. Okay? And I have a picture on the next slide explaining this. Things within passive transport are diffusion, facilitated diffusion, osmosis, and filtration. So they just happen on their own without any input of energy. And the rest of the lecture here kind of digs into each one of these things. We're going to explain what diffusion is, facilitated diffusion, osmosis, and filtration. And then the same down here with active transport. So with active transport, you have to have energy. Okay? So it requires that input of ATP. So then we're going to talk about active transport pumps, endocytosis, and exocytosis last. So this is going to be an example of me telling you passive versus active diffusion here. And so active requires energy. Look, the guy is having to use energy or ATP to push it up the hill. But in passive, it takes no energy. That ball is just rolling down the hill, correct? On its own without any help at all. So within passive, so this is the ones that require no energy here at first, okay? The first one is diffusion, okay? And this is going to be the most common form of transport. This is the most common thing that happens. There's a movement of a substance from an area from a high concentration to a low concentration. So the picture there on the left, picture A there, they're dropping a red tablet into the water. And so that tablet is very concentrated, right? Like medication you're taking. All the medication is within that tablet. It's very, very concentrated. But if you put it in the water, it's going to start off from a high concentration in that tablet, and then it's going to dissolve to the picture like 3 and go to an area of low concentration because then it's all spread out. Also within diffusion is going to be an equilibrium, which is the point at which concentration is equal. Equilibrium, equal. This is also showing you diffusion within passive transport as well, but more specific to the body. And this is talking about the oxygen exchange versus carbon dioxide within the lungs, more specifically the alveoli, which is where the gas exchange happens. Okay, so this is saying if you're starting off with a high concentration of oxygen, and then you end up with a low concentration of carbon dioxide. So that's a good thing. Facilitated diffusion. So we've talked about diffusion and now facilitated diffusion within passive transport. Within facilitated diffusion, there needs to be a helper, okay, a helper molecule that assists with the movement of the substances from an area of high concentration to low concentration. So we're still in passive transport. We're doing high concentration to low concentration, but this requires a helper molecule for the cell. So something like glucose or sugar, right, is absorbed from the intestine by facilitated diffusion. So it has a helper molecule. So another way of passive transport, we talk about diffusion, facilitated diffusion, and now osmosis. Some of you have maybe heard of osmosis before, but I'm going to explain it, and especially with this picture here a little bit. You've heard of it, but you don't know what it is. Good. So diffusion, osmosis is the diffusion of water through a selectively permeable membrane. So in some circumstances what this means, and especially, this is really a specialized case of diffusion, is osmosis. So because the solute, which is, say, the water, okay, say it's water, here in this picture. Because the solute is unable to move, water is diffused from the compartment where there is more water to the compartment where there is less water. Right? So, to make it even. So, through osmosis, the water is moving, but the substance, the solution is moving, okay? The solution is the water in the picture. But the red things, which are the particles, okay, or the substances, they can't move across that membrane. Does that make sense? So the water moves, but the substance does not. An example of this is going to be, you have a patient with a high sodium level, okay, or a high salt intake. And so say you give them some more, they already have a high sodium level, they're already maybe swollen a little bit in their lower extremities, they're having swelling, and this isn't from like an injury or anything, they're just carrying water in their lower extremities, their feet, their legs, so forth, okay? But you keep on increasing their salt, or you give them some type of solution that includes salt, then you are going to create this problem even worse, okay? Because the water's still going to move and then create more swelling, because the water's going to move, but the substance of salt is not going to move. And then you're just going to have more and more and more. So then that's where we get into these different types of solutions. And we'll get there in just a second. The term tonicity means the ability of a solution to affect the volume and pressure within a cell. And so then we need to discuss isotonic, hypotonic, and hypertonic solutions going into this. So iso means equal. So this solution is equal. It's the same concentration as the intracellular fluid. Consider a red blood cell as placed in the solution because the solution is isotonic and there should be no movement of water occurring. Okay? The cell's not going to either gain or lose any water because it's trying to keep things equal. It's an isotonic solution. Hypotonic, okay, hypo, so under tonic solution. If the red blood cell is placed in pure water, which is a hypotonic solution, okay, then the water moves into the cell by osmosis. The pure water is more diluted than inside the cell then. And so, so then it is considered to be hypotonic or under. We never give pure water, straight water, through an IV because it would cause this imbalance. Right? Because water is considered that. Hypertonic is going to be one of those solutions that does not, or it causes the red blood cell to shrink because it is placed in a very concentrated salt solution. All right. So the water diffuses out into the red blood cell, into that really, really heavy salt. Imagine an ocean, okay, with that solution in the red blood cells. So I am going to let you watch this video here, and then I'm going to be watching, I'm going to be looking over and looking at yours. I may have this too loud. They're testing next door, I think.
Before we take a look at how osmosis and tonicity affect the cell, let's review what each of these terms means. Osmosis represents the diffusion of water across a semi-permeable membrane. The term tonicity refers to the relative solute concentration of two environments separated by a semi-permeable membrane. In other words, by comparing the tonicity of the solutions, you can determine the direction in which osmosis will occur. To demonstrate how tonicity affects a cell, let's place some red blood cells into a beaker containing pure water. In this case, the solute concentration is greater inside the cells than in the surrounding water. In other words, the contents of the cells are hypertonic in relation to the hypotonic contents of the beaker. Because of this, osmotic pressure results in the diffusion of water across the membrane and into the cells. Over time, if enough water enters the cells, the cell membranes may burst. This is called lysis. Now let's place the red blood cells in a beaker containing a solution of salt, such as sodium chloride. Since the contents of the beaker are hypertonic in relation to the interior of the cells, the water within the cells will diffuse across the membrane and into the contents of the beaker. This causes crenation, or shrinking of the cells. If the red blood cells are placed in a beaker whose contents match the tonicity within the cells, then there is no net gain or loss of water. The environments within the beaker and inside the cells are said to be isotonic, or the same. An important thing to remember is that osmotic pressure always causes water to move from a hypotonic environment toward a hypertonic environment. In other words, water moves toward areas of high salt or sugar concentrations. This simple process is used to drive the operation of our kidneys and explains some of the physiological consequences of diseases such as diabetes.
Okay, so going back to the lecture here. So that was over osmosis and tonicity and that water moving basically and where the water was going to go move in a salt situation or a sugar concentration. So we were working on passive transport before and we're done with that, and then we're going to move on here to active transport.
Oh my gosh.
I know. Active transport requires what? Active, you're being active, so when we're being active, we have to have what? We have to have energy, okay? We have to eat. That's why Colton can eat so many snacks, because he needs the energy for his game tonight, right? So with active transportation versus passive, you don't need any energy. Active, you need energy, okay? You need nutrients to get some energy. You have to eat. Active transport pumps, and it moves substances from an area of low concentration to high concentration. So exactly this difference, or the other way than passive transport, because passive went from high to low, Where active, you need energy, and it takes you from low to high concentration. Okay? But it requires energy. More specifically, what it includes is endocytosis, exocytosis, so we're going to talk about that now. With endocytosis, this is taking in, okay, inside. So the ingestion of taking substances by the cell membrane because the particle is too large to move across the membrane by diffusion. So it has to take it in by eating, or phagocytosis, that's cellular eating, or pinocytosis, which is cellular drinking. Okay? So an example, what? 10 more. White blood cells, don't be rude. White blood cells, bacteria help take up the bad stuff for us. That's what they're there for. Okay? White blood cells are there to protect us from illness.
We call them the soldiers.
That's right. The white blood cells, yep. So then the next active transport mechanism is going to be exocytosis. Exocytosis is the secretion or getting rid of, secreting of cellular products out of the cell. So getting rid of the things that are unwanted in the cell. An example here is going to be when the cells of the pancreas make proteins for use outside the pancreas, but then they push them out to get used, okay, by someone else. So they use part of the proteins, but then they push them outside the pancreas to get further use after that. We're not going to watch that video. And then one of the last things here that we're going to be talking about for a few slides is going to be cell division. And there's a few things here at the end, but cell division. So this is when we kind of go back to that mitosis that we talked about and meiosis, which we did talk about earlier. So cell division is necessary for the body's growth, repair, and reproduction. You need your muscle cells to regrow. You need your red blood cells to produce more every day, every 5 seconds. You need them to reproduce. Okay? But then some of your nervous cells, they never reproduce. Okay? Once they're gone, they're gone. So the frequency of cell division varies from one tissue to the next. Some cells reproduce frequently. Red blood cells are going to be the most frequent. Okay? That more than 2 million red blood cells replaced every second. Every second. And certain nerve cells do not reproduce at all. Mitosis is where there's bodily growth and repair. It is the splitting of one mother cell into 2 identical daughter cells. Where meiosis, it's the sex cells only. Okay, it's the sex cells only. The cell cycle itself is going to be the sequence of events the cell goes through from one mitotic division to the next. And so that's what we're going to talk about here a little bit more. So we're talking about the mitosis, the reproductive phase of the cell. Okay? There are 2 phases of the cell cycle, the interphase and then mitosis. And then within the interphase, it is divided into 3 groups. I'm not going to take it any further and read off these next 3 things to you because I don't expect us to go this far in the physiology. Okay? Just need to know that the cell, there's 2 phases of a cell cycle, and it's interphase and mitosis, cellular division. During the mitotic phase, the cell divides into 2 cells. Look at that picture A up there. Okay? During the mitotic phase, the cell divides into 2 cells in such a way that the nuclei of both cells contain identical genetic information. So the picture on to the left there, that's, say, a mother cell, and then it divides into 2 daughter cells. But they both have genetic information. Within this phase, though, the mitosis phase, there are really 4 phases. The prophase, the metaphase, anaphase, and telophase. The chromosomes line up, and they split into 2 identical sets. And they just continue splitting, and continue splitting, and continue splitting from there. These 4 phases of mitosis, again, I'm not going to go into great detail or a lot of detail about them because I don't think that it's all that important. I just clicked back. So just remember that a cell divides into 2 cells, and the nuclei of both cells contain identical genetic information. Oh, I think I'm going to skip this one because what that video did is it really, I watched it this morning, it just repeats itself over and over and over again, and it's 5 minutes of repeating itself in many different ways. So there's that.
What were the repeat?
What?
What was it?
I'm talking about mitosis, just the same thing over and over again. And I can just tell you from turning you blue in the face about it, mitosis is you start off with one and then it separates into two genetically identifiable cells, okay? That's the main thing you need to get from that. So, cell differentiation. This is when we talk about cells that there gets to be so much reproduction that they kind of start to change in their shape and their size, and this is when we get unhealthy cells, or things like cancer cells, okay? But it also can create specialized cells as well. So, some of this can happen too much. So, like red blood cells, nerve cells, muscle cells, they all look and function differently. But we all start life as a single cell that divides many times into identical cells. At some point, the cells start to specialize and differentiate by switching in enzymes that produce red blood cells, bone, or nerve cells. Cancer cells are often called poorly differentiated cells. And then, has anyone ever heard of stem cells? Stem cells? So it's pretty newer in the past 10, 20 years or so. This is how they created the dolly, the lamb, the sheep. You know what I'm talking about, Dolly the sheep. So it's like it was a sheep created from science, not from natural, like reproduction. Okay. So they took stem cells, or they specialized cells that are transplanted to form cell to bone marrow, skin, adult cartilage. So some of these things would be really helpful if we could recreate cartilage for somebody, right? If their cartilage was all wore out. Yeah.
I heard that they're gonna start putting lab-made beef into stores now.
Oh yeah. Oh yeah. I think it's already there.
There was some lab-made beef, and it doesn't really have the nutrition that you would
Typical beef does.
get from a rotisserie raised cow. But it's disgusting.
I don't know.
It's not made right. I mean, cattle are grown the way we grow it for that. And you're not getting what you're supposed to.
Yeah. It's just, what are they having to use to supplement to get some of those things? That's what's scary, like chemicals and things like
I
that.
think we should all just turn to vegetarians.
Okay. So stem cells do present an ethical dilemma because it's almost as if we're creating life. Does that make sense? And so stem cells really can be an ethical dilemma. So this is showing the picture of what I just described, that we all start out as an unspecialized cell, and then it breaks off into the specialized cell, the neuron, the red blood cell, the muscle cell, bone cell, and then epithelial cells, which are going to be our skin cells as well. This means when you start to see disorders at the end of a lecture, that means that we are getting close to the end, okay? So atrophy. Atrophy happens with your muscles. That's the most common thing I think about. Atrophy happens if you're not actively using it. And I don't mean exercising every day, but if all of a sudden I'm working, I'm going home, I do this, right? I get up every day, I brush my teeth, I take a shower. What if we happen to be an animal or a patient that we got in a wreck and we broke our leg, and that's all, but we require hospitalization because of a major leg surgery, okay? And we lay in a bed for 7 days. What happens?
You forget how to use that leg.
Okay, yeah, those muscles really start to waste and atrophy. They break down. Okay, so, and they're not rebuilding that, those cells and stuff as quickly. And so this really can lead to wasting away, even in a short amount of time. And so that is why they can come in your room, and that's why we're going to do range of motion and physical therapy, like somebody said. But also, so this is a decrease in the size of the cells. It starts at the cellular level, but then goes all the way to the tissues and organs that we have to live. The term dysplasia. This is a change in the shape, size, or organization, and this is when it can become cancerous, those cells. If they are dysplasia, they don't look right, okay? And this is when they become cancerous. Hyperplasia, this is when there's too many or an increased number of cells. So this could also result in an increased size of a tissue or organ. So your liver is way too large, or your heart can become enlarged because of different reasons, but mainly because of hyperplasia as well. Then there's metaplasia, where it's a transformation of one cell type into another. And then necrosis. When a patient has or person has necrosis or frostbite, that means that that's tissue or cell death. Okay, that's completely dead or gone. That's what the term necrotic or necrosis means. It is cell death or group of cell death is tissue death. Lastly is a neoplasm. A neoplasm, what that means is it's an abnormal new growth, usually a tumor. Okay, that's the fancy word for a tumor. And what happens is there's malignant neoplasms and there's benign neoplasms. So which one are cancerous, malignant or benign? Malignant or benign, which ones are cancerous? Malignant. Remember, mal, bad, benign, not. Okay? So, malignant neoplasms are going to be those cancerous tumors, and they are invasive and tend to metastasize or to move from the original primary site to other parts of the body as well. And that's talking about how cancer spread, basically. Okay? So, because those cells are still dividing even though they're not healthy, and we just got to find a way to make them stop. Okay? So, this is the end of lecture for chapter 3.